Optimal health for high performers. This is the Health Upgrade Podcast with Dr. Nawaz Habib.
Hello, everybody. Welcome back to the Health Upgrade Podcast. I'm here with JP. How are you, sir?
I'm doing very well, thank you.
This episode of the Health Upgrade Podcast is brought to you by Truvega. Have you been looking for a simple way to feel calmer, think clearer, and sleep better? Imagine achieving this with just 2 minutes a day. I'm excited to introduce you to Truvega, a handheld device that supports your nervous system through gentle vagus nerve stimulation. As a doctor specializing in functional health and wellness, I've seen firsthand how Truvega has accelerated healing in my clients. I've used this device to help people with a range of issues, from sleep disturbances and heart palpitations to digestive dysfunction, nervous system imbalances, and mental wellness concerns. The results have honestly been remarkable. Truvega is designed to catalyze and accelerate the shift in your body from fight or flight to rest, digest, and recover mode, helping you manage stress, maintain mental clarity, and unwind more easily. It's not just theory. I've witnessed its positive impact on real people facing real health challenges. The best part? Truvega fits seamlessly into your routine. No need to carve out hours for wellness. Just 2 minutes a day can make a difference. It's a small investment of time for potentially significant benefits in how you feel and function each day. If you're curious to experience Truvega for yourself, visit truvega.com/upgrade. You can save $30 on your device by using the code UPGRADE30 at checkout. Remember, that's truvega.com, truvega.com/upgrade. It might just be the first step towards a calmer, clearer you, just as it has been for many of my clients. We're going to talk about the brain today, and we're going to talk about it in a very particular way. We're going to talk a little bit about how the brain becomes excited and how signaling is increased in certain areas of the brain or sensitized. And we're going to talk about how it's slowed down and calmed. And almost to this point of what are some of the neurotransmitters, what are some of the chemicals that are present within these brain tissues that essentially push the accelerator and push us into an excited state or an excitable state? And what are some of the tools, neurotransmitters, that we can use to shift towards pushing the brakes and slowing it down and getting kind of the brain into a calmer state? And how do we create a balance between these two where we have moments where the accelerator is being pushed and moments when the brain is being pushed? I think this podcast episode is going to be really good for anybody who's interested in brain health and understanding how the brain functions and how inflammation plays a very, very important role in this balance between accelerator and brakes, or the excitability and inhibition of brain signaling. So please share this with anyone who you think might be interested in this particular topic. But why don't we get into kind of the basics right off the bat? Let's talk a little bit about, when we talk about the brain, we cannot really dig into it without truly understanding nerves, neurons, and neuronal connections, how these brain cells fit together with one another, and what other cells and signals are necessary for maintaining and ensuring that these signals are passed effectively through these nerve cells. So why don't we start off with some basic neuroanatomy?
Sure. I think it's a really great place to start. So if you think about the connections in the brain, we've got 86 billion neurons and something on the order of at least that many other cells, whether they be astrocytes or oligodendrocytes or microglial cells, that are playing a role. And I always talk about the fact that the microglial cells are sort of the conductors of everything, which makes the brain really an immune organ, because microglial cells are the tissue-resident macrophages of the brain. But the nerve cells have really three major parts to them. They have the cell body, which is sort of the typical thing that all cells have. That's where the nucleus is. That's where a lot of the organelles are, et cetera. Then nerve cells are specific in their structure in that they have a series, typically a large number, what are called dendrites, basically. These are protuberances that go out and make connections with other cells. And then they bring signals into the cell body area. And then there's one axon. And there are subsets of neurons that have more than one axon, but by and large, there's one axon. And that is what the nerve fires or with a firing of a nerve is sending a signal down that axon. And so the way it works in the connections that are set up is that there's a series of other axons from other nerves that are connected to the dendrites of a given nerve, the nerve we're talking about, nerve A. And all of those other nerves are sending signals, and the dendrites of nerve A are picking up those signals. And if those are inhibitory signals, then that message goes down to the cell body and says, don't fire. Or they can be excitatory signals, and those signals will come down and they'll tend to push that cell body towards depolarizing and firing. And so basically it's a summation of all of those different signals that are coming in from the dendrites into the cell body that determines whether or not that cell is going to fire. And if it decides that it's going to fire, then it has an electrical discharge that sends a message down the axon. Now that axon is going to have, it's typically only one, but it is going to make many connections. There's going to be lots of different other cells, downstream cells, that are going to connect. And the connections are made, again, through synapses. So you have the axon giving off a presynaptic terminal, and the other cells that are downstream, other nerves downstream, their postsynaptic terminals on their dendrites are picking up that signal. So the closeness of those two presynaptic and postsynaptic terminals forms the synaptic cleft. Now, around the synaptic cleft, there are other cells. These are specifically the astrocytes. And astrocytes play a really important role, sort of as the maintenance crew, as well as the controllers. They're like the people in the control room tuning the volume, if you will, of that connection. Now, microglial cells also come along, and they're constantly, they spend about over 90% of their time, I think it's 94% of their time, testing whether or not synapses are functioning properly. And if they are functioning properly, then, and they're needed and necessary, then they will reinforce that. And what reinforcement really means is that they change the structure of that connection so that less neurotransmitter needs to be released in order to activate that synapse and cause the dendrite to pick up a signal.
Essentially lowering the threshold, correct?
Yeah, exactly. It makes it easier. It's sort of like that old story about a bike. If you ride a bike through the forest, there's no path, but if you do it 1,000 times, well, then there's going to be a path. And that's sort of what we're talking about. If that synapse is firing regularly, then we want to make it easy. We want to make it something that happens with a low threshold.
I think the reason for that from a biological standpoint is that we don't want to require so much energy in order to create the effect that is being created, especially in a reinforced functioning connection. So essentially, think of it like you meet a new friend. You're going to put a lot of effort into creating this new friend initially. And it takes a lot of effort to decide, am I going to text this person? Am I going to be in connection with this person as often? And slowly and surely, as the connection becomes good and strong and you have this positive relationship, you can text this person out of the blue without truly even putting the effort into thinking about it. Same kind of idea is happening here. And that ease of signaling, the ease of being able to communicate between these two cells is happening because of these microglial cells making it easier to do that. I think that's just a simple analogy to understand this process.
Yeah, they're giving off chemical signals basically that tell the nerve and that synapse to change how it releases the neurotransmitters, how close the synapse is, and how the astrocytes function to maintain that synaptic cleft. Again, it's the importance of those tissue-resident macrophages of the brain, the microglial cells. So what we're talking about right now is whether or not that signal that's released by the presynaptic terminal is going to be an excitatory signal or it's going to be an inhibitory signal. And then the effects of and how the astrocytes function and how the microglial cells function in different circumstances. And we talked about inflammation being a really important one, shifting the balance of how much excitation there is versus how much inhibition. So let's dig into the two of the most important neurotransmitters in the brain are really related to one another very closely. It's glutamate, which is an excitatory neurotransmitter, and GABA, which is an inhibitory neurotransmitter.
I would even go as far as to say as that glutamate is the excitatory neurotransmitter in the brain, and GABA is the primary inhibitory neurotransmitter in the brain. That there are other neurotransmitters. We all know about kind of the fancy, really well-known ones, the dopamine, the serotonin, the acetylcholine, because we're the vagus nerve people.
Norepinephrine, orexin.
Exactly. We have tons and tons of these, but glutamate and GABA are the primary ones that essentially create excitation or push the accelerator on the glutamate side. And GABA, which is the primary inhibitory, which is kind of the initial pushing on the brakes. So let's talk a little bit about how glutamate works, how GABA works, and how the connection of these two is actually understood. What is that close relationship between them?
Well, in both cases, the reason why I said they're very related to one another, they really all come from the same amino acid, which is glutamate or glutamic acid. Those two are really basically the same depending on the pH that they're in. And what happens is that neurons have the ability to take glutamine, which is, again, one of the forms of the amino acid, and turn it into glutamate and then release it through vesicles into the synaptic cleft to then bind to receptors on the postsynaptic terminal. And so that's how it functions. Basically, the nerve is firing, signals going down the axon. That triggers the release of these vesicles, these little bubbles, if you will, that contain glutamate. And they fuse with the cell membrane at the presynaptic terminal. And as they sort of merge, think about the bubble sort of opening up and releasing the neurotransmitter into the synaptic cleft. That is all being monitored by the astrocytes, because after that release and after the effect on the postsynaptic terminal, you got to clean it out so that it's ready to standing by to send a signal or not. And that clean out happens by the astrocytes taking up that glutamate. And typically under normal circumstances, that astrocyte converts that glutamate back into glutamine and then returns it to the nerve cell upstream of where that synapse is, so that it's not really interfering with what's happening at the synapse. And so that's the cycle. The synapse releases the glutamate, the astrocyte takes up the glutamate, and then converts it back to glutamine, gives the glutamine back to the nerve cell turns it back into glutamate, so it's ready to be released again.
I think that's an important thing to understand. It's just cycle of activation, deactivation almost of the glutamine amino acid into glutamate. That is important. Now, glutamate is that primary accelerator. So what is the effect of glutamate when it does get to that postsynaptic membrane, when it enters the dendrite of neuron B in this case? And what is it then going to say or do to the postsynaptic neuron? What is it going to say to the next cell to do?
It tells the postsynaptic neuron it alters calcium levels, it alters ion levels, and as a result, it causes the dendrite to send a signal up into the cell body of the downstream neuron that says, hey, I'm going to tell you to fire. You're going to listen to all the other dendrites to see what they say, but this channel is saying fire.
Yeah, this is essentially activating the signaling pathway. It's saying we need to send more signals. We need to tell more people. We need to alert the other neurons of something. And we need you to send this signal more powerfully, more effectively, and very quickly. So it's a very rapid thing, the glutamate activation.
Yeah, think about it. It really does lend itself to sort of a digital circuit, because basically you're either telling it's either on or off. That's why these neural networks are really very close approximation to how a brain works, because the brain is basically sending a signal that's either turn on or turn off. And you've got 10 different connections or 100 or 1,000 different connections that are all sending signals either saying turn on or turn off. And that is then summed. And literally the cell body acts almost like a little counting machine. And it says, if I've got 1,000 dendrites and 501 of them say fire, well, then I'm going to fire. But if 501 say don't fire, then I'm not going to. And so even you could have a positive signal from one dendrite always firing and always on. If it's not got similar signals coming in elsewhere, it's not going to cause that next downstream cell to fire.
Yeah, absolutely. Let's go to the other side of this now with GABA. So you mentioned that there's a close relationship. How does glutamate turn into GABA? What's the kind of dropped it right here, but glutamate and GABA are so interconnected that it's literally that GABA is produced from glutamate.
Yes, that's exactly right. There's a synthesis pathway that is actually GAD, is the glutamate.
I believe you said decarboxylase, right?
I just can't remember off the top of my head what it is. I think it's glutamate decarboxylase, GAD. Yeah, I don't know where the A comes from.
Let's see if I can find it. Yeah, it's just glutamate decarboxylase.
Yeah, I just don't know why they would have added the A in there. But in any event, glutamate decarboxylase exists in two isoforms, basically GAD 65 and 67. And they basically create this new molecule, GABA, which is gamma aminobutyric acid. And that serves sort of the opposite. Instead of sending the digital signal 1, it's sending the digital signal 0. It's saying no need to fire. Now, it's interesting because you would think that all nerves have, there's only a need for one receptor type. GABA receptor is going to be activated by GABA release. But there's actually a couple of different types of GABA receptors. There's GABA A and GABA B receptors, and they have slightly different functions and slightly different places where they're expressed. I mean, it's important, but for our topic today, I think focusing on how nerves and how the release of these neurotransmitters shifts and how nerve function shifts and how astrocyte function shifts based on inflammation is really the goal here of this discussion. So I think it's time to reintroduce that really important cell type, the microglial cell, and say, how does the microglial cell and its activation state impact the expression of neurotransmitters? And we've seen this before because we've talked about it with respect to serotonin and dopamine. And I think those people who've heard those episodes and heard us talk about this are going to be very familiar with this role that inflammation plays.
Yeah, absolutely. Let's talk a little bit about some of the clinical findings or the things that people will notice in their day-to-day lives that might show up when we are out of balance. So when I talk about out of balance, I'm talking about if we have a little bit of glutamate activation and we have a little bit of excitation, it turns on certain neural circuits, it turns on certain neural pathways, it's exciting to those brain circuits, it's essentially pushing the accelerator for a small component of the brain. And a GABA activation is going to push the brakes and slow that same circuit down if we were to do it on the one particular circuit that we're talking about. But there, I imagine, are moments, and we talked about inflammation being one of those major indicators that's going to push us in one of those directions. Which direction does inflammation push us in? Is it more towards glutamate or is it more towards GABA? I know the answer, but I want to allow you the opportunity to dig into this.
Yeah, inflammation is a huge trigger for excitation. The brain is aflame, and it drives a lot of excitation and the synthesis at all levels, both at the neuron level, at the astrocyte level, even at the microglial cell level. The microglia themselves can release glutamate. So it is an all-out war at that point. When the microglial cells say we're inflamed, glutamate levels rise.
How quickly can that happen?
It can happen very quickly, especially in areas where you are processing pain. Pain is one of the 4 cardinal signs of inflammation, along with redness and swelling and heat. Heat obviously coming from the swelling and the blood flow and the redness. But there's areas of the brainstem that are responsible for receiving these pain signals and then interpreting them. And how they interpret is really a function of the level of inflammation that's happening in the brain itself. So anytime there's inflammation in the body, there is a trigger for inflammation to occur in the brain because it's bidirectional. And so what you see is in animal models, in fact, there's a great study that was done by Michael Oshinsky, who was at the time, he was in Philadelphia at Thomas Jefferson University. He's now at NIH. But his study, he had established a model of pain in which he had sensitized animals so that when they were exposed to a specific type of trigger, they would experience significant pain. And the way he showed how that pain was being experienced was that there was a very sharp rise in glutamate levels on the order of minutes. So if you can imagine, there's two types of animals, one that's been sensitized and one that hasn't, and he's measuring the glutamate levels in this area of the brain, the trigeminal nucleus caudalis, and measuring the levels of glutamate that were expressed. In the normal animals, they're relatively stable. There might be some level of increase associated with the trigger, but it's modest. I mean, it's less than doubling. Whereas when you expose the animal that's been sensitized to the same trigger, you can see upwards of over a 90-minute period, something on the order of 7 to 10 times more glutamate. So you're talking about a 700 to 1,000% increase in glutamate levels. And even over a span of 15 minutes, you can see a double or tripling. So it's a very rapid process, and it all appears to be triggered by an inflammatory response, because the way you sensitize the animal is to expose the animal to repeated inflammatory triggers, and then you trigger an inflammatory event to acutely.
What are some of the more common tools used to create that inflammatory event in animal models? Because they're based on things that humans experience and things that are happening maybe on a lower grade level generally, but in certain cases it can be high. But are they using things like lipopolysaccharide, LPS? Are they using other inflammatory triggers as well?
So if they do it peripherally, the answer is yes. You can use things like lipopolysaccharide, which is an element of a bacterial membrane that is very, very inflammatory. I mean, our bodies recognize it instantly. There's also something called poly-IC, which is used as a sort of a mimicking of a viral infection. These things are very, very inflammatory. You can use other things. There's Freund's adjuvant and things like that. But at the end of the day, in the brain, what he was using was, he referred to it as an inflammatory soup, but it included things like bradykinin, prostaglandins, and other things. Subsequent to his doing the study that I was referring to, he did boil it down and say, listen, prostaglandins are really all you need. And prostaglandins are obviously something that we express when we're injured. We have those in our bodies, and they respond to traumas and inflammation, triggers very rapidly.
So clinically, what somebody experienced symptomatically, or what would somebody in practice notice in their patient when they're experiencing this excessive activation of glutamate and essentially this sensitization creating kind of a pain experience? What are some of the more common either conditions or particular symptoms?
The model was a model of migraine. So what we were showing was that the activation of this sensitization pathway that caused this rapid increase in glutamate was similar to what occurs in a person who's having a migraine. And people who have migraines, they oftentimes have some sort of prodromal symptoms that are telling them, hey, something's going on here. Something's changing in my brain. I'm going to be interpreting this differently. One of the phenomena that people who have migraines often experience is called aura, which is a visual disturbance, so sort of a blind spot or a fuzzy spot or some sort of discontinuity in their visual field. And that's a sign that there's what's called a wave of depolarization or a cortical spreading depression. So when you see it in your visual field, it's in the back of your head, it's happening. But there's a spot where all of the nerves are sort of in a synchronized wave depolarizing. And that leads to inflammation. There's some reason to believe that it's a signal that the brain in that area was deprived of oxygen, not necessarily enough to kill it, but enough to trigger a hypoxic signaling. And what basically, getting into the weeds here, but that cortical spreading depression, there's still a lot of discussion about what its purpose is. But one of the purposes I think that people are coalescing around is that when a small area of the brain is deprived of oxygen or experiences some level of hypoxia, it sends a signal to the rest of the surrounding area and the tissues, and it can go all the way through the brain in its entirety if it's strong enough, telling those cells, be prepared you may need to exist with a lower level of oxygen. And that affects mitochondria. It activates glycolysis. And when the mitochondria feel this, get this sense of stress, there's a strong trigger to create inflammation. And so the pathway appears to be that this cortical spreading depression is triggering an inflammatory response. The inflammatory response then leads to this increase in glutamate. And in the areas of pain perception, there's people have mood changes, behavioral changes associated with cognitive processing changes that occur associated with this prodromal experience, this aura experience. And then what they end up having is a very severe pain response. And that pain response is associated with heightened levels of glutamate being released.
Is that something that people also that have been diagnosed with fibromyalgia are often experiencing, or chronic pain syndromes, things like that?
It's been associated with those things as well. There's a lot more complexity associated with fibromyalgia and some of the widespread pain conditions. But yeah, the mitochondrial dysfunction is at its heart, a part of so many different conditions. From fibromyalgia to chronic fatigue to mood disorders and neurodegeneration. So yeah, it's a very important thing.
So when we're in this inflammatory state, essentially when the glutamate is upregulated, when there's a lot of this glutamate that's been increased to the rate of 7 to 10x or 700% to 1,000% increase in glutamate, and we have this cortical spreading depression, and the microglia are all going into this inflammatory process state, and astrocytes and neurons are all hyperfiring, and we're in this hyperexcitable state.
And CGRP is being released, and it's changing blood vessel permeability. And now you're getting extravasation of fluid into the brain as a result. Yeah, it's a hot mess.
Yeah, it sounds like it is. And I imagine in that hot mess, something is lost. And I think the thing that's lost is, how do we turn that down? How do we inhibit or slow down this excitatory activation of glutamate? What's happened to glutamate decarboxylase, the enzyme that turns glutamate to GABA, in this particular situation?
Well, it's being inhibited. It's TNF alpha and the other inflammatory cytokines are blocking that enzyme. And as a result, you're not making anywhere near as much glutamate. But it's even worse than that, because that inflammatory signal does a few things to astrocytes as well. We talked about it, when they're functioning properly, astrocytes are taking up the glutamate. So they're sort of cleaning up, they're part of the cleanup crew. They're cleaning up the glutamate, they're turning it back into glutamine, and they're giving it back to the neuron. When there's inflammatory signaling, as these microglial cells, as the conductors, they're telling the astrocyte to stop doing that. And what the astrocyte actually does is it takes up the glutamate and then it spits it back out. So you end up with the astrocyte actually contributing now to the problem. And it's not just the GABA is being downregulated. The GABA receptor populations are being downregulated. So you've got the presynaptic terminals releasing glutamate. It's being triggered to release the glutamate. The astrocyte's not taking it up. It's actually adding more glutamate to the situation. Frankly, so are the microglial cells. They've got the ability to release glutamate to just continue this excitotoxic state. And the presynaptic terminal, the postsynaptic terminal, which has the GABA receptors on them and is hoping to get some downregulation, it's being triggered to actually have fewer GABA receptors expressed. So GABA A and GABA B receptors are being downregulated. And that postsynaptic terminal is being promoted to have more GABA receptors. So your NMDA receptor populations are increasing as a result. So it really is a cycle, a vicious cycle. It just continues to progress. And that's why migraines sort of continue to get worse and worse and worse. Now, we talked a little bit about mitochondria in this situation. And one of the things that's really interesting is, and relates to that hypoxia theory about how the cortical spreading depression start. And one of the things that appears to be very positive, especially for severe headache conditions like cluster headache, is high concentrated oxygen. So you see a lot of patients who have cluster headaches, they're almost tethered to these high flow oxygen masks that are giving them liters and liters, like 12 liters per minute of oxygen, in the hopes that there will be a high enough oxygen concentration that they can actually push the mitochondria to function more effectively.
Is this the mechanism by which hyperbaric oxygen therapy works?
It very well may be. I mean, there's lots of different pathways that are activated, but that in fact may be one of the really important ones. You want to shift back into a more healthy energy production, oxidative phosphorylation with healthy mitochondria, and downregulate the dysfunction of the mitochondria and the reliance on glycolysis.
Yeah, absolutely. So we're in this cycle. Let's say, for example, a patient is stuck in this chronic inflammatory cycle. The glutamate activation is generally on the higher end. We have less ability to turn on GABA. How do we break this cycle of inflammatory, excitatory activation? How do we get out of this? And I think we'll talk a little bit, both of us can talk a little bit about how the approaches that are showing up now and things that have been around for thousands and thousands of years can actually help to shift this. But what are some of the ways to break that inflammatory cycle in the first place?
Well, obviously the microglial cells are your conductors of this entire inflammatory cascade. So they're sort of an obvious target. But stepping away from them just for the moment and just talk about some other, because we got involved in headache, some of the other treatments for headache. We talked about the fact that CGRP is released. It's calcitonin gene-related peptide. It's one of the most potent vasodilators. So it's dilating the blood vessels, allowing that fluid to come out, allowing inflammatory cells or inflamed cells to enter into the brain from the bloodstream.
The mechanism by which it does that, just quickly, is through activation of the iNOS, the inducible nitric oxide synthase enzyme, which is the inflammatory version of nitric oxide synthase. And what that does is that's exactly how it creates that extreme vasodilation. It literally causes the blood vessels to almost become leaky and open up extreme levels of that vasodilation.
Yeah, and that's why the triggers that are used to activate in the animal models are typically NO donors. So, try not to, basically like TNT, but nitroglycerin and things like that are very strong triggers of this. But, so before we get to the microglial cell directly, one of the things that's been proposed as a treatment for headache is to prevent that dilation. So things that are really potent vasoconstrictors. The very first drugs that were developed for headache really were ergots and ergotamine and things, and dihydroergotamine, which are very, very potent vasoconstrictors. And they have lots of potential risks, cardiac risks, et cetera, because they're working systemically and they're altering your vasculature pretty profoundly. Subsequent to that, there are triptans, which also have an interesting cardiac side effect, which is not so great. That's why you shouldn't take triptans like they're, all the time. You shouldn't take them more than, a few times a month, but people do. And they're working on the blood vessel side of things. Then there are preventative drugs that try to limit the ability of this cortical spreading depression to spread. Things like epilepsy drugs, like topiramate, and things that are actually end up being GABA analogs. They don't actually function like GABA, although they look like GABA. Things like Neurontin and gabapentin. These drugs were originally developed as anti-epileptic drugs to prevent that sort of excitatory, overwhelming of the brain, that storm that occurs of activity. But at the end of the day, if you can control the inflammatory signaling, the inflammation that's being triggered by the microglial cells, that really appears to be very, very effective, both in the long term, preventing the cortical spreading depression, preventing the blood vessel dilation, and allowing the brain to remain in a more balanced state. It's a very it's a very potent target. And of course, I think what you were referring to, is we oftentimes refer to, is how do you get away from that excitotoxicity associated with glutamate overexpression by downregulating inflammation? You can do that by activating the parasympathetic nervous system and activating those microglial cells to move back into a non-inflamed state and downregulate the levels of TNF-alpha. And it can happen very rapidly. That's the part that's so fascinating, is that all of this can be regulated within minutes. by vagus nerve stimulation or parasympathetic activation.
Yeah, and there's stronger ways to do it using tools like vagus nerve stimulators such as Truvega, or utilizing tools that have been around for thousands of years like breath work, right? So I think this is an important thing for people to understand, is that the cycle that is inflammatory and excitatory and seemingly very challenging to break can be broken. And it's important to note that there are tools that are available to help break that inflammatory cycle and shift the body to a more of an inhibitory state, or more of a calming rest, digest, recover state. I initially wasn't leading in this particular direction, but we were obviously going to get to this because the effect of vagus nerve stimulation is on regulating these tissue resident macrophages throughout the body, but in the brain in particular, those microglial cells. The target of vagus nerve stimulation is microglial deactivation and putting it into not a deactivated, turned off sleep state, but a regulatory state where it can manage and monitor and conduct good business and maintain the balance of glutamate and GABA within those synaptic areas, and astrocytes being obviously another target cell of these tools. And it's acetylcholine that is the neurotransmitter that drives this particular activation. Acetylcholine is a whole other neurotransmitter produced from acetyl-CoA and choline, two very important nutrients. Acetyl-CoA is very heavily linked to metabolism and mitochondrial function. And here we come back to the mitochondria again. And choline, which is a very important essential nutrient that we do not get enough of in our bodies. It's generally, our diets are actually about 90% deficient in choline. And so what we want is to have these tools available so that we can then utilize the vagus nerve to turn on those signals, to get to those cells, and to regulate these immune cells. I think that's an important thing to bring up.
It's amazing how these episodes that we do connect together. We recently recorded an episode about cholesterol, and one of the villains of the cholesterol myth, if you will, were eggs. And one of the best sources of choline are egg yolks. So, and even within the egg, the thing that they vilified the most was the yolk.
The yolk.
So it just makes no sense. Everybody out there is having egg white omelets and saying, no, I still have eggs, but you're not getting the choline. The choline is in the egg yolk. You have to have that. And the cholesterol that's there is really good for you also, by the way. And so at the end of the day, you're right that we're deficient in choline. And one of the reasons why we're deficient is because we were told not to eat, things that have a lot of cholesterol. And the problem is that they also have a lot of choline. And so we need, the good news is we're busting the myth of vilifying cholesterol. And we're showing that that choline sources are actually quite good for you, both for the choline and for the rest of the nutrients that they have. But you're absolutely right. Acetylcholine is critically important. And tying it back to the mitochondria, we've talked about this before as well. The function of mitochondria is in large measure dependent on what the acetylcholine levels are in around the cell and within the cell. And they have on them, the mitochondria have on them receptors for acetylcholine that help them to function better. And it ties back to melatonin, and there's a relationship between melatonin and the expression of this receptor, which is the alpha-7 nicotinic acetylcholine receptor. And we should at some point do an episode about nicotine and how important nicotine actually is in your diet. And people are saying, nicotine in my diet? Yes, it's present in lots of foods. Eggplant, for example, and cauliflower and other things have lots of nicotine. And so we'll talk about that at some point as well. But getting back to just, GABA and glutamate, one of the studies that Michael Oshinsky did was look at how vagus nerve stimulation could affect that dramatic increase in glutamate levels. Because he had already shown that if you sensitize the animal with inflammation, as we talked about before, you could get that 700 to 1,000% increase in glutamate expression over a relatively brief period of time. And that it tracked with the level to which the animal experienced pain. So as the glutamate levels went up, the pain levels went up as well.
So very clearly a dose-dependent response. The more glutamate, the more more activation, the more pain experienced or the more symptomatic nature.
Exactly. And so what we did was we looked to see whether or not vagus nerve stimulation could reduce the levels of glutamate and whether or not that would tie into a lower level of pain. And so the first study that was done was, okay, let's stimulate the animal and then expose them to the trigger. So basically almost like a prophylactic treatment. And what we found was that if you did that, if you dosed the animal with some vagus nerve stimulation prior to the exposure, the animal never experienced that 700 to 1,000% increase in glutamate, and the pain levels never got higher than what they were at baseline. So it really had the ability to completely prevent this. But I then asked the question, I said, that's wonderful scientifically. But if you're going to use vagus nerve stimulation to acutely treat a headache, then it would be helpful if it could do that even though it had already begun to rise. It could stop the glutamate level rise or even reduce it back down to baseline. And that's a tough thing to do because what you're asking the therapy to do is do something very rapidly after a cascading process had already started. It's one thing to block you got a rock that's going to roll down a mountain. It's one thing to prevent the rock from rolling down because you got a little wedge there, okay? But once the rock starts rolling, it's a lot more difficult to stop it. So it was a tough test to see whether or not it could work. But it was absolutely remarkable. And I remember sharing this with some of the top leaders in headache when we finally got the data and we were looking at it. And I remember at the time, the president of the International Headache Society, he looked at the data and he stopped. He said, that is powerful. That is interesting. Said that you could take, and what we saw, so that everybody can get a visual of this, the control animal, whether you trigger them or not, they're going to maintain basically a flatline level of glutamate. So it's a normal baseline level. The sensitized animals start out looking just like the normal animals, but when you trigger them, they begin to see an increase that relatively linearly increases from the same level up to about 7 to 10 times that level of glutamate over a period of 90 minutes. And we were looking every 15 minutes. So we were measuring the levels of glutamate in that tissue every 15 minutes. And what we found was that if we let it go for the first 60 minutes or so, or even 45 to 60 minutes, so we had 400 or 500% increase in glutamate, and then stimulated the animal's vagus nerve, that we could drop the glutamate levels back down to baseline, so indistinguishable from the control animals, within 15 minutes. And it was within the next measurement. So I don't even know. It could have been shorter than that. It could have been even more rapid.
But it happens within that 15-minute.
It happens so rapidly. And the reason for that, I believe, is that the change in the levels of TNF-alpha are extremely rapid. The vagus nerve stimulation affects the microglial cells very rapidly. And that's interesting because TNF-alpha has a half-life that's actually pretty long compared to 15 minutes. It's actually long enough that it should be still around. I think that there's actually a mechanism for retaking that TNF-alpha back up. And so it's actually reabsorbed. And again, that's yet to be confirmed, but it makes sense given the half-life of TNF-alpha. And what you see is an immediate shift. Once that signal is not there, pushing that inflammation, I think the astrocytes almost immediately begin shifting back to taking up the glutamate and not resending it back into the synapse, but the astrocytes actually convert it back to glutamine and send it back to the neuron. I think it's a longer process to change the postsynaptic terminals expression of GABA A receptors, GABA B receptors, and DA receptors. I think those populations change more slowly, which is why you typically, I think, have some, I'll call sort of a post-migraine hangover, post-migraine brain fog, if you will. I think there's still some level to which the postsynaptic terminals expression of receptors has to change. But I think the glutamate levels clearly come down very rapidly, and I think it's the astrocytes and the nerve itself that are changing their levels of expression rapidly.
Essentially what they're doing with the glutamate then.
Yep.
Is there a link to the GABA in that particular process? Do we note that as the symptomatic nature of these pain processes decreases, is there an increase in GABA
yes.
As well?
There is, and it's both in GABA expression, largely in the periaqueductal gray, which is an area of the brain that's associated with pain, and other things as well. What we also see, and this has been shown in studies with epilepsy patients, is that over a period of time, and that time may be weeks or months. In fact, I think it's even 6 months to a year out, the density of GABA receptor populations goes up. So you end up seeing, the lower levels of inflammation will shift your GABA, specifically GABA A mostly, but you see an upregulation of GABA A receptors. And GABA A receptors are really important for pain modulation.
Yeah, absolutely. I think understanding that when the accelerator is heavily pushed, trying to push the brakes right off the bat may not be enough, right? We're going to burn through the brake pads a little bit. We're going to have a little bit of burnout. And this is where you're trying to do certain anti-inflammatory processes, but the inflammatory process is so strongly and rigidly activated that it's hard to get out. And so sometimes we can initiate some practices and not see the payoff as quickly as we were hoping, or almost minimal payoff at all in terms of not experiencing the reduction in those symptoms. Versus if we hack this system, and I'm doing this air quotes, hack the system, meaning utilize the vagus nerve to rapidly initiate the changes to those immune cells present in the brain, in the central nervous system, microglial cells, the astrocytes, to turn down the amount of glutamate that's being released, turn down that glutamate activation. Essentially, we're lifting the foot off the brake, then the brake, or off the accelerator, lifting our foot off the accelerator, then the brakes can start to work more effectively. They don't need to work quite as hard. And so that's where the GABA activation will work.
Yeah. Sometimes, anybody who drives a car on a highway knows you don't necessarily have to step on the brake in order to slow down. You can just take your foot off the accelerator and the air resistance will slow your car down. I think that the interesting thing about vagus nerve stimulation in this setting is what you're doing is you're changing the thresholds for being sensitive to something that would trigger you. Again, in the animal models, we can create the animal models to be robustly sensitive. I mean, very sensitized, very reliably sensitized. Human beings live their lives in a variety of different ways. It's not a controlled environment. And so every one of us has a threshold for some level of a trigger. I always say, I don't typically get headaches. I'm fortunate that way, but that doesn't mean I can't get a headache. I'm quite certain that if I drank enough alcohol and I stayed up, late enough and didn't get myself enough sleep, and maybe somebody, knocked me in the head, I will get a headache. I will have a migraine. I mean, you could cause me to have that, but it takes quite a lot to do that. That means that for me, I have a high level of GABA A receptor population, GABA B. I have a balanced level of glutamate and GABA in my system. I have robust mechanisms for downregulating inflammation. But there are things that you can do in your life that will slowly degrade those things. And it doesn't happen overnight, but you will slowly degrade them. Yes, getting sick, having inflammation, those things will lower your threshold, will bring you to a higher sensitization level and make you more susceptible to headaches. And so one of the things that the headache neurologists will do is they'll talk through lifestyle changes and food, because food sensitivities can be a big trigger for this as well, and show you how you need to rebuild your resistance or lower your susceptibility to these triggers. And over time, vagus nerve stimulation can be a very powerful way to assist you in resetting where your threshold levels are so that you are much more resistant. That's why it's been given an FDA clearance for treating migraines preventatively, because what you can do is you can downregulate your potential for getting a headache. So now, and one great study that was done over in Italy with this was in women with menstrual migraine, because something on the order of 2/3 of all migraines are experienced in some measure menstrual or menstrually related. And what they showed is that those shifts in hormones are actually part of the sensitization process. And that's why the age where migraines typically penetrate into a person's life is actually around puberty. The highest level of penetration is actually around age 14. So showing is that hormonal shifts are part of that process. And by lowering the inflammation levels, you can shift that level down so that you're less likely to get those headaches when the hormonal triggers hit.
I think it's also really important to note that when the threshold can be lowered due to various reasons. You can have genetic susceptibility. So if your parents had experienced something like migraines, there may be a genetic component to the lower threshold or the higher sensitivity piece initially being there. But again, this is an important piece. Genetics are not the cause of all of these things. Not all of these things are genetic conditions. It can be environmental. It can be habitual by your family as well. You have a point there. Go for it.
Yeah, I was just going to say about the genetics. There was a population or genome-wide study that was done, and one of the really remarkable findings was that migraine headaches are associated with genetic, let's say, phenotypes that are associated with inflammatory conditions. So that's why we find headaches associated with autoimmune diseases and chronic conditions of inflammation. So what we're seeing is that there is a direct connection between migraines and headaches and other pain conditions and this chronic level of susceptibility to inflammation. It can be genetic, but you were going to say it's also...
I think there's a lot of environmental, exactly. And so dietary is huge, right? We know that there are certain food triggers for migraines. We know that there are certain food triggers for a lot of inflammatory conditions. Some people are sensitive or to the point where they're actually allergic to gluten, right? We have gluten sensitivity. We have dairy sensitivity. There's a lot of people that can't handle nightshade vegetables because they will trigger an autoimmune type of condition. So we know that there may be
part of the leaky gut syndrome.
It's all linked, right? They're all absolutely linked, and then certain proteins can make their way in and mimic proteins within our body, creating that autoimmune type of reaction as well. Leaky gut syndrome is a big component to, or the leakiness of your gut will contribute to the low-grade constant inflammatory process. And if you have a lower threshold genetically or due to other susceptible reasons that you are more sensitive to this inflammatory reaction, then a little bit of low-grade inflammation triggered, generally if you're sitting close to that threshold and you eat some gluten or you have a tomato and you're sensitive, it pushes you just above that threshold and all of a sudden, snap, here we go down that cascade, right? So one of the major things that we can do is use dietary protocols, just like is done by functional medicine practitioners and a lot of these functional diagnostic nutritionists and all of these wonderful practitioners, naturopaths, even some very good medical doctors will change your diet, will make recommendations to eliminate certain foods. Often we'll start with a full elimination style diet because what we're doing is we're trying to lower the amount of low-grade inflammation that's currently present, right? So that way we're further from that threshold to begin with. And vagus nerve stimulation can increase the threshold. So essentially we're trying to separate these two things. For those who are on video, you'll not see, but I'm using my hands to show that we're creating this increased space or this larger gap between the amount of inflammation that's present and the threshold required to create that excitatory glutamate response. And so this is where the lifestyle factors, the exercise factor is going to be a huge one in this.
I was just going to play with that and talk about.
Let's get into that. Let's talk a little bit about how exercise reduces inflammation, because if there was a pill that was anywhere near as powerful as exercise is, it would be the most effective pill for inflammation ever known to mankind.
It would be the anti-aging serum.
Period. Yes.
I mean, it is absolutely critical. And the reason for it is that exercise, at the highest level, exercise is a hormetic stress. So you're basically stressing your body and stressing your system, including your autonomic nervous system, in a way that's voluntary, typically. And that exercise then makes you more capable of handling other stresses. And remember, your immune system doesn't care whether the stress is voluntary, involuntary, physical, emotional, mental, chemical. It doesn't matter. It's going to respond exactly the same way. And so by inducing small stresses, and exercise is a really great way to do that over a short period of time. It's very defined. You just step off the treadmill, put down the weights, you're done. That is a great way to increase your thresholds, increase that separation between where you live and where you exist at baseline and where your sensitivity threshold is for triggering, some pain condition like migraine. That's one part of it. But at the deeper level, it's also about your mitochondria.
I was just going to get to that point.
Especially endurance exercising. Your body, your cells need to have the healthiest mitochondria. They need to have the most mitochondria that cell type, can have. You want to be in a state where oxidative phosphorylation, respiration in this way is easy to do, okay? And is something that you're flexible. You're not reliant on one fuel source like sugar. You want to be able to move into a state where you can, use fatty acids and ketones and other things easily and fluidly. So you want to use exercise as a trigger because there's, very few triggers for creating more mitochondria in your muscle than use. It's something that everybody knows because everybody who's ever eaten chicken or had, duck or, poultry knows you got white meat and you've got dark meat. Dark meat is dark because it has more mitochondria, okay? White meat is tissue that is not being used. So that's why if you have a chicken, chickens that are raised to be, eaten as poultry, they're not flying around.
They're using their legs.
They're using their legs to walk around. So their drumsticks and their thighs are filled with mitochondria. That's their dark meat, but they're not flying. So the breast meat is white. It has very few mitochondria. That's why you want your fat. That's why people do cold plunges and they do things to stress their fat cells into being, thermogenic, generating energy. And they do that and you turn your white fat into beige fat. And ultimately you want to have, brown fat, that really the healthiest type of fat. The coloration is associated with mitochondria. And how do you get your cells to produce more mitochondria? How do you get your mitochondria to reproduce? Well, that signal is simply from use. If you go out and you exercise, you're using that tissue, you're using your muscles, you're using it in a way that demands more energy be available. And so your body responds. It produces more mitochondria.
I don't think there's a more true statement when it comes to mitochondria than if you don't use it, you lose it. And use of muscles and use of these tissues to activate mitochondrial activation, to activate the muscle to require more energy and thus produce and signal to have more mitochondria present is literally the way that we turn on mitochondrial presence, and repetitive use increases the efficiency of these mitochondria that are present as well.
And the relationship between your nervous system and your mitochondria is fascinating. In fact, I had the wonderful honor to speak with a really brilliant researcher just yesterday. And he's focused on the consequences of sort of catastrophic injuries like paralysis. And what you find is that when the nervous system is no longer connected to the musculature in a way that's controlling it, so you have an injury. Now the muscles in your legs, you have no voluntary control over them. What happens is there is a catastrophic loss of muscle tissue because there's a loss of mitochondrial function. And you say, well, and I asked the question, it's like, well, suppose you use electrical stimulation like TENS units or other muscle activators to activate that muscle. He said, listen, it can help, but it is in no way, shape, or form going to give you what's simply the tonic level of your autonomic nervous system and your voluntary muscle control of that tissue gives your muscles. There's going to be catastrophic loss of mitochondria and as a result, muscle cells in that tissue. It's nearly unavoidable. And his focus is to try to figure out ways that we can get that tissue to remain active. And we talked about the potential use of acetylcholine as a trigger for keeping those mitochondria healthy. So it turns out that it's not just under inflammatory conditions, it's under situations where there just simply isn't the signal.
Yeah, less acetylcholine literally just being sent, period, tonically in a baseline level. I think that's so important to understand. We talk about acetylcholine in the context of the vagus nerve all the time. Acetylcholine is the neurotransmitter of the neuromuscular junction. How motor nerves speak to muscles is only through acetylcholine. So movement and vagus nerve function literally use the same neurotransmitter. This is, I think, two sides to why we need to A, turn on the vagus nerve, and B, provide acetylcholine effectively to those muscles so that we can maintain their optimal function and utilize them and exercise them. And if we don't use it, we literally are going to lose it. It's insane to say this.
We don't just lose it from a health standpoint, physical health. We lose it from a mental health standpoint, because one of the things that we now know is that muscles, when they contract, they're releasing molecules. They actually call them hope molecules. They are very positive for your mental health.
These are the endorphins?
No, it's not just endorphins. It's actually a whole class of separate molecules called hope molecules. And they are fascinating to study, and they play a role. But at the end of the day, the nervous system, mitochondria, muscles, as you said, the neuromuscular, but the immune system, there's these chat cells. Chat cells are releasing acetylcholine. So acetylcholine is, we decided that when we were going to discuss this, that we were going to pick two neurotransmitters that were really, really, really important. We were going to pick glutamate, which is your primary excitatory, and GABA, which is your primary inhibitory. And where do we end up? We end up in an organic conversation talking about acetylcholine. So
it really goes to show that no neurotransmitter works in a silo by itself. They all are working together. We didn't talk a lot about dopamine, serotonin, norepinephrine, norexin. Like, we didn't talk about all of the other neurotransmitters, of which there are many, but it really does come down to control over immune function, control over mitochondrial function. And I think obviously we have a little bit of a bias towards acetylcholine, but you can see the power, you can see the effect of it in numerous different ways, because when we don't utilize acetylcholine effectively, when we don't produce it effectively in good quantities, and we don't have the signaling capacity in the nerves to be able to send it out, we have inflammatory issues that become uncontrolled. And that is the root of a lot of health conditions. The vast majority of these chronic health conditions that people are suffering from are due to a lack of inflammatory control. And what we need is control over inflammation. And the only true powerful way to create inflammatory control is through acetylcholine. So we'll always come back to it, but note that glutamate and GABA don't work in a silo, right? Serotonin and dopamine don't work in a silo. They're not in a vacuum. They're all interacting with one another. And there are certain things that need to happen to initiate improvements in the balance between all of these things. And vagus nerve function, acetylcholine are so powerful. I'm just going to quickly shout out our sponsor for this episode, which is Truvega. And I think this tool is really, really powerful. It's one that you work to develop along with a team of wonderful people. And Truvega has graciously been our sponsor. Yeah, I've got mine right here as well.
I'm never too far away from this. I know how important acetylcholine is. I know how important it is to keep my inflammation levels down.
I love it. I have one at my desk here. I have one at my bedside, and I have one in my daughter's room because we use it with her all the time. So I'm the same way. I'm never too far from my Truvega either. So this is a great tool, but breath work is a really powerful way of doing this. Mindfulness, meditation practice, humming, chanting, gargling, singing, all the things that we break down in both of our books. These are actually really powerful ways to get your body into a situation where it can manage immune function.
Yeah, I refer to it as parasympathetic recovery mode. You need to get into that state. It's fine to be in a sympathetic state, especially if it's voluntary, for short periods of time. I always say, you can go out and run 5 miles, and that's good for you. Go out and run 50 miles, not so good for you. Go out and ride 500 miles, and you'll be dead. Okay, so you got to get rest. It's fine to do 500 miles in 2 months. It's not so good to do it in 2 days.
We don't all have to be David Goggins.
And look at listen, David is an amazing human being. Really,
100%.
truly amazing. But the damage that he has sustained and the damage that he is going through, I mean, my understanding now of his knees it's catastrophic. You don't need to do what he's doing. In fact, you shouldn't do what he's doing. I understand he's taking it to the extreme to make a point, but you will injure yourself. You will cause yourself problems if you try to live his lifestyle. And frankly, he shouldn't. He should dial it back to what's healthy, not what's extreme.
Yeah, and I fully agree with you on that. And for each person, it's going to be a little different, right? To the point where you're not breaking your body down, you're actually working to build it up. And yes, there's a little bit of breakdown with exercise initially, but the intent is to create a resilient body because you're bouncing back as effectively as you are. So whether it's 20 minutes on the Peloton or 8 miles on the treadmill or what works well for you, go and do that. Go and do that exercise. Create these habits of positive change to activate muscle tissue, to activate vagus nerve signaling, to activate this parasympathetic recovery mode that we talk about. Because what it's going to do is it's going to help support that inflammatory control. It's going to help downregulate the glutamate. It's going to help upregulate the GABA activation and create balance between these two very important neurotransmitters that we've discussed today. And it's going to just allow your body to recover from the day-to-day knockdowns and the big traumas that your body is inevitably going to experience. This is what we're always talking about, is how to upgrade your health is truly to initiate that recovery response, to learn how to turn on your ability to recover from stress. I think that's a great way to kind of wrap a bow on today. Any final thoughts from you, JP, there?
No, I think that's it. You just said it perfectly. Just the goal here is to make you more resilient. And the way to think about this is the lower your thresholds are for moving into that pain, the less resilient you are. The more resilient you are, the higher those thresholds are, the larger the gap is between where your baseline is and where you're going to start to experience symptoms. And you can do that a variety of different ways: exercise, diet, sleep. We always talk about sleep being important. We didn't spend that much time on it today, but all of these things tie into how you can modulate your autonomic nervous system to make yourself healthier.
Yeah, 100%. I think that's a great way to wrap up our wonderful discussion today on excitatory and inhibitory neurotransmitters, primary ones, and how we always take our little tangent and talk about acetylcholine and vagus nerve stuff. But I think this is a really important one for people to understand. And so I want you to, if you got to this point in the podcast, congratulations. You heard a lot of science. You heard a lot of positive recommendations. And I would love it if you could share this with one person who you think could utilize this information to help positively upgrade their health. Thank you so much for listening. And JP and I will catch you on the next one.